how can we go from the genetic information that is encoded in the DNA to the actual phenotype, so what a plant looks like, how it behaves under stress conditions or during its whole life. In this population, about every 40 base pairs of the DNA, there is one which is different between those lines. What people found is if you switch on this gene in a very strong manner, then plants are very drought resistant.
If plants have the potential to be immortal, then why do they die? Welcome to Apple French Pudding, your gateway into the world of science. Today's scientist is Julia Engelhorn, a junior professor in the Genomics of plant adaptation to climate change at Université de Montpellier. Today we will be discussing the Booster project, where she's trying to boost drought tolerance in key cereals to mitigate the effects of climate change-driven drought. Welcome, Julia.
Hi there, Jeroen. Thank you so much for having me on the show. It's really nice. I'm really looking forward to our conversation.
I'm looking forward to it as well. But before we delve into it, do you have a fun science fact for our listeners?
I'm not really sure if it's a fun fact or more like a kind of very interesting fact or something exciting. It's at least something which kept me excited about plants, and that is that plants are potentially immortal. And that is because they have these structures which are called meristems, which are actually pools of stem cells that the plants retain throughout their lifetimes. And you can see these cells actually in action when you go into your garden and, for example, you cut a branch from a tree. It will grow back, but you would never try that with your own fingers, for example. So there's a big difference between plants and animals. And this kind of also leads to some plants being thousands of years old. So there are these trees or rose bushes which have been around for thousands of years and seen so much of history. And that's something I just, find fascinating and maybe a little bit funny.
That is amazingly fascinating. So if plants have the potential to be immortal, then why do they die?
So I think some plants just have that programmed in their life cycle somehow. So there are these plants which just live for a year. They have a pre-programmed life cycle. And at one point, they kind of consume all these meristematic cells. And they use them, for example, to make flowers and fruits and give that to the next generation. But some plants do not, and then they can live for a very long time.
Okay, well, that's very cool. So, but today we will focus more on plant genomics. You have dedicated years to plant genomics, and I am curious, why are you drawn to this topic? Was there some special event that triggered this interest for you?
Yeah, there were a few events, and they are also related to the stem cells and the meristems a little bit. I kind of got this idea to talk about that. So when I was a child and I was dog walking in the forest, I very often saw in the springtime, you could see a lot of new trees sprouting from beech nuts and acorns. And I was always fascinated how kind of from these little small structures, you could make a new tree. In the beginning, I didn't even know that these were baby trees because I just saw these plants and they look completely different than the trees. And then I saw the beech nut or the acorn still stuck on top of them, and I realized, oh, this is a baby acorn tree, oak or something like that. Of course, then I didn't know why that is the case. I just found that fascinating. And then when I went to university, I learned that this is because of this very different strategy of plants to live their life. So they're not born with all their organs, but they develop their organs throughout their lifetime. So when I would come back the next week to the forest, then they would already have 2 more leaves. And then these leaves would already look a bit more like the real leaves from the trees. And this is all because they're constantly producing new organs from these stem cells. And so I got very, very interested in these structures, these meristems. And then I think the most crucial impulse came from my PhD supervisor, because she gave me the opportunity to work on a project which concerned the genes which are working in the shoot apical meristem. So that is the part of the plant which is making all the aerial organs of a plant. And so during my PhD, I could study the genes which are regulated in the shoot apical meristem and how they are regulated. And I got the chance to study these things with genomics methods just at the starting point when these new methods were really taking off, and they were really starting to transform our knowledge of gene regulation. And yeah, since then I've been basically hooked to the question of how genes are regulated.
Yeah, because it's also interesting, like you said, the first two leaves of a plant sprouting are totally different compared to
Yes.
the leaves when it's adult, basically.
Yes, in the beginning, I couldn't understand that, how this is actually possible.
Yeah. So you said you studied the shoot apex. So that's the tip, actually, the top of the plant, right? Or of the branch.
Yes, exactly. So that's the very top of the plant where new leaves are starting to develop, or at one point this can change and then it can make flowers, and depending on which state of the development the plant is in.
Okay. And so you studied this, and you studied this with genomics. But what is plant genomics, or what is the difference, for example, between plant genomics and traditional genetics?
In general, genetics and also genomics is interested in the fundamental question of how we can go from what is called like a genotype to a phenotype. So how can we go from the genetic information that is encoded in the DNA to the actual phenotype, so what a plant looks like, how it behaves under stress conditions or during its whole life. And the difference between genomics and classical genetics is more like that in genomics, we're looking at the whole genome. So how are things regulated on a whole genome scale? But in traditional genetics, it was more like people were looking at like, how does one gene or one pathway with a few genes influence these kind of characteristics?
And if I'm not mistaken, in your research, you check genes, but also non-coding regions. What are those?
Yeah, so non-coding regions, first of all, let's maybe define what coding regions are. That makes it a bit easier.
Yeah, makes sense.
So on the DNA, the genes are the parts which are encoding proteins. So they code for proteins. They contain basically the instruction to build a protein. So, and when we're speaking about the genetic code, we talk about the language in which this kind of instruction is written. But when whole genomes were sequenced, we saw that only around 2 to 20%, depending on which organism, which type of plant, of the genome is actually genes. So only 2 to 20% contain these instructions. And the rest of the genome is what we then call the non-coding part, because it doesn't contain such an instruction. It doesn't code for a protein, but it rather helps to regulate genes and maybe have instructions on when and how often a gene can be switched on.
Okay, but that's very interesting. So actually, between 80 and 98% of your DNA is actually non-coding.
Yes.
Okay, and so that actually the majority of your DNA, and that is being used to regulate the part that is being coded. Is that correct?
Yes, to more or less, it's not that all of the parts are equally important of the non-coding genome, and that's what makes it a little bit hard to study. But yes, so this part regulates how the other part, the coding part, is actually used.
Yeah, because there's also something like junk DNA, or that's a term. I think that's outdated now, but maybe you can give us some insights on that, on the relation between like junk DNA and non-coding regions, because that's not the same, right?
Yeah, so the thing is that, yes, the word junk DNA is a little bit outdated, and it started in the beginning when scientists first sequenced genomes and they saw these numbers and they were like, what, 2%? So what is the rest? And so then they didn't really know what the rest was doing, and they saw that, oh, maybe this is just some evolutionary leftover, some junk which is still there but doesn't have a function. Of course, at this point, scientists already knew that the regions which are very close to genes are useful and are important to regulate the genes and to switch them, to help switching them on and off. That was already known. But they saw that all the rest of the genome, they basically called that junk DNA at one point. But now we are more and more discovering that more parts of this, what used to be called junk DNA, is actually also important for regulation and for other parts, like for structuring the DNA, for example. And so we are learning in this field, and we're discovering more and more parts, and we're discovering parts which are regulating genes, although they are very, very far away from the gene that they're regulating. But we still don't have like this universal understanding of how genes are regulated through non-coding regions as we have for the genetic code, where we know exactly, okay, these bases in the DNA, they code for this part of a protein. We don't have that for the non-coding part yet, but we're understanding more.
Okay. Because like the typical idea of junk DNA that has no function, I think evolutionary, that would make no sense. It would like be removed, right? Because if it has no function, why would we sustain it?
That's what people were wondering, because making DNA, and DNA is in every cell, and every time you make a new cell, you have to replicate the whole DNA. And if you would do that with a large part of the DNA, which you don't use, that wouldn't make any sense exactly. So somehow, even the parts where we don't know what they're doing, I think we just haven't found their function yet.
Okay, so now we have some idea of what non-coding regions are, and that's actually the majority of our DNA. But you put a lot of research in it, if I'm not mistaken. And why do you think those regions are so interesting?
Of course, in the beginning, when people were looking at how to improve, for example, traits of plants, so to make them more resistant to some, or resilient to some stress, or to produce more fruit, people were focusing on genes, because genes were known, genes were well characterized, and we know they control proteins, and they are very important for all the different functions within the plants. But that was successful in a few cases. But at the moment, we are more and more discovering that it's actually the control of genes that still holds this enormous potential to make smart plants. So to make plants which are, for example, more resilient to stress. And so why do we think that there is this potential? And that's because of some recent studies where we have looked at how nature actually enables the adaptation to changes, and what nature has already provided us with in terms of differences in these non-coding sequences. And they found that about half of these natural differences that are responsible for trait differences, so for example, making a plant more resistant to something, are located within these non-coding regions. So there's a huge potential. And if we would only look at the genes and the coding regions, we would miss half of the DNA differences, for example, between different plants, that could actually explain us why, for example, one maize plant flowers earlier than another. So these non-coding DNA regulators are very important for the timing and for kind of ensuring that proteins are there at the right moment, and that has an enormous potential.
Okay, so like a lot of phenology, like the timing of when everything happens, when a plant flowers, when it fruits, when it grows even, is determined by those non-coding regions.
Yes, exactly. So because you can imagine the genes are always there, right? So they're the same in all the different cells, but somehow the plant needs to know, okay, when do I need this gene to make a flower, and these kind of things. So they really need to be tightly regulated somehow. Okay,
so when people are breeding plants to flower earlier, they are actually breeding plants with a different non-coding region.
So far, this hasn't been exploited so much. I mean, it has in some cases just empirically, because people found like these alleles, like these differences, which are kind of non-coding, but it hasn't been like structurally done like for genes, for example, because for genes we know, okay, these genes are important for flowering time and we can do something about that. But for the non-coding regions, we kind of lack the ability to find exactly those places. And they have a very high potential for breeding, because you can imagine that you can make smaller changes than what you can do when you would really mutate a gene, for example. Because if you would make more of a gene the whole time. So you can imagine, for example, a gene which is like conferring more drought resistance, or more drought resilience to a plant. And with this gene, for example, a breeder could say like, okay, great. So let's put this gene on all the time and really, really strongly. So then we would have super drought resistant plants. But what people found then are super like disease resistant plants. But what people very often found in these cases where they had like these finally more resilient plants, they found that in the end, when they're looking at the plants in normal conditions, because it's not always that a plant gets infected. It's not always that there is drought. These plants have like enormous disadvantages. So they grow more slowly. They have like less yield somehow. And you can imagine that this makes sense because they are producing a protein the whole time, which they do not need in these conditions. So, and that's where these non-coding regions become so interesting because they could help us to switch on these genes only when they are needed.
Yeah. So, yeah, in general, that's something that I have encountered as well. So when plants are bred to be more drought tolerant, for example, then they are more tolerant to drought, but they have a lower yield. And if you're able to use these non-coding regions, you can switch on and off certain parts so that it's drought tolerant when it's needed. So it doesn't produce things when it's not needed and the yields are higher under good circumstances. Is that correct?
Yes, exactly. I mean, at least that's the theory and that's what we're trying to find.
Yeah. Okay. So, and your work, you use these genomics, plant genomics, to study the effect of drought on plants. And can you give us some insights into your work?
Yes, of course. So I can tell you a little bit about how we try to find these kind of elements. So we are really after these elements which only, for example, switched on under drought and switching on a beneficial gene. So, but that means we first of all, we have to look at this vast space of non-coding DNA, and we need to find which parts of the genome are actually interesting or which could harbor these regulatory elements. And for this, we had to go in several steps. So first of all, we needed a way to narrow down the sites which could be really interesting. And one way that we did that is looking at the binding sites of transcription factors. And transcription factors are proteins which are important to regulate how and when a gene is actually read and is used. Because we said the whole time, okay, these non-coding regions, somehow they regulate how genes are read and how genes are used. But how do they do that? And actually, most of the times they do that with the help of transcription factors. So these proteins bind to these regions, and then these proteins can, for example, recruit other proteins, which would then read out the DNA and start making the templates for making the proteins. And they could maybe also make some loops in the DNA to bring certain parts of the DNA together to really tell them, okay, now when all these parts come together, then please switch on the gene. So these transcription factors are the starting point of our research of how we want to study drought stress. Okay, I didn't start about drought stress yet, so that's a bit of a longer story. But first of all, that was our idea how to narrow down a little bit this place in the genomes. So yeah, how do we go on from there?
Yeah, well, that's a good question. Yeah, please go on. Because now we have heard, so you have these transcription factors which can actually bind and then determine when things are being read or not. How do we proceed from that? Because I think this is also the work from the Booster project, if I'm not mistaken.
Yes, exactly. So that's where this big consortium where I'm a part of comes in. So that's a consortium which I'm part of because of the previous lab I was the lab I was previously working in. So in Cologne and in collaboration with the University of Düsseldorf. So it's a large consortium of a lot of different research groups in Europe and in Africa, and also some breeding companies and some seed stock locations. And we are trying to find these elements in the genome which can really be beneficial for switching on and off genes for drought stress improvement.
Yeah. So these transcription factors, actually.
Yeah, these transcription factor binding sites. So the sites in the genome which are important for the binding of transcription factors. So how do we get that together somehow? So we have this method which was developed in my previous host lab together with another group in the US, where they find a way to look at where all the transcription factors in a plant genome are located at a given moment. So before that was very complicated to look at that because there were experiments which were only for one transcription factor. So you would have to do one experiment and then you would get one transcription factor which would maybe bind to like a few genes, maybe from like a few hundred to a few thousand. But then there are like around more than 2,000 transcription factors in maize. So if you would have to do like 2,000 experiments to see where all the transcription factors are sitting. So,
That's a
and
lot.
then, yeah, that's a lot. That would keep a lot of scientists busy for a long time.
Yeah, I can imagine.
So yeah, so the idea that they had is like, what if we have like an indirect method? And so they came up with this idea that transcription factors can leave some footprints on the DNA. And how can they do that? So they use like an enzyme, which is a bit like a Pac-Man. So it likes to eat DNA, but it only likes to eat DNA when there is nothing else bound to the DNA. So you throw this enzyme on the DNA, it eats away all the DNA except for the places where a protein is sitting on the DNA. And then what you get in the end is all the places which were protected because a protein was sitting there. That's not only transcription factors, there are also large proteins on the DNA, which assure that the DNA is nicely packed within the nucleus of a cell. But the other ones, like the smaller parts of that, are transcription factor binding sites. And so we can select these small fragments, and then with a lot of new technologies, so this what maybe some people have heard of, like next generation sequencing technologies, we can read out all these little small fragments and get the sequences of them. And so in the end, we do some computational analysis, and then we know exactly where these transcription factors were located in the genome. And then now we know that for all the transcription factors at once, because we didn't have to look for specific ones, the enzyme works for all of the transcription factors. I still haven't told you about drought stress, correct?
No, but I'm just going to reframe so to make sure that I'm following. So if I'm not mistaken, so you have the whole genome and you have an enzyme. The enzyme actually likes to eat the DNA unless when there's a protein on it. And that's, for example, a result from the transcription factors or a transcription region. And so those regions remain and other regions as well. And now you actually, because of the enzyme, you only have the regions you want that are left. And then you can read which they are. Is that correct?
Exactly. That's correct. We have already narrowed down the whole non-coding space to these transcription factor binding sites. That's already a big, big improvement. So we don't have to look at the other sites anymore. But then what you would do with classical genetics is like, because now you have a binding site, but there are other things apart from the DNA sequence which are actually regulating if a transcription factor is binding there or not. There can be chemical modifications on the DNA, or there could be like a stronger packing of the DNA. So we don't really know if the sequence is actually responsible. So to find that out, normally what you would have to do is you would have to mutate each single site. So you would go and you would exchange some of the sequences of the DNA, and you would see, okay, when I destroy this kind of binding site, then we have less binding. So that means the DNA sequence is actually really responsible for that. But again, it's even worse than with the transcription factors. There are like hundreds of thousands transcription factor binding sites. You could never do that. But we are quite lucky because nature has done that for us in a lot of cases already. Because especially, for example, in maize, where we're working on a population of maize, and within this population, about every 40 base pairs, so every 40 letters of the DNA, there is one which is different between those lines, at least in some of them. So we have this really huge diversity in nature, which we can use, and then we can look at, okay, if we're now looking at transcription factor binding sites in several different maize lines, then we can see if there is a difference in the sequence, there can be a difference in the binding. And with that, we can find the places where the DNA sequence is actually responsible for differences in binding. And then we can come to the drought.
Okay. But so just you're using now the natural variation in different strains of maize, right?
Yes, exactly.
Yeah, and that's why you use maize in this project, because it has those strains with those different DNAs, or sequences that you can use?
Yes, so we used maize for several reasons, because there was a lot of information already available. So we had a lot of sequence genomes where we knew those sequences. And of course, maize is a very important crop to study, and it has a lot of problems with drought stress. Even now in Europe, that we're unfortunately seeing more and more. And yeah, there were huge populations available. For example, in the booster project, we're using a European maize population, and we have like 30 different maize lines where we're looking at these transcription factor binding sites.
Okay, because I checked the project, and you're not only using maize, you're also using Teff, I think. Why Teff then?
Yes, exactly. So Teff is like a very, very interesting plant. So it's like a plant which is not so well known here in Europe, but it's very, very popular in Ethiopia and Africa. And it's a very drought resistant plant. So for that, it's quite interesting to us. And it's a superfood. So it's like very healthy. And so it's a really interesting crop to look at. And I mean, we have to kind of think of new ways for agriculture with climate change. And so Teff was this kind of really great opportunity to look at that. And that's why we're so happy that we have like University from Ethiopia within our project, and some experts on Teff from Switzerland, which are helping us with this project. And so we're also looking at the diversity in these in the Teff, to figure out if we can find some regulatory elements in Teff, because Teff is already very, very drought resistant. But first of all, there's always room for improvement. So maybe we can find something to maybe combine some of these sites and make Teff more drought resistant. Or we are also looking at, and that's something which we do with some collaborators in Belgium, they're also looking at if we can transfer some of these sites which we can find in Teff, if we can transfer them to maize, actually.
Okay, so we're narrowing down, right? We've gone from the whole genome. We extracted those regions where the transcription factors actually are occurring or attaching. Now we have natural variation in maize and in Teff. Now we need to go to drought. What is drought? Because it's a very broad term, right? How do you define drought, for example, in your project?
Yes. That's a very, very good question. And it's very broad. That's true. So because we want to have like a very controlled environment, we define drought as only a short time where we withheld watering completely. So we had plants growing all in a randomized manner in a greenhouse from all the different lines. We watered them all the time until they reached a certain developmental stage. So they had like 4 leaves, for example. And at this point, we divided them, and one half of them didn't get any water anymore, and the other ones were still watered normally with their normal regular schedule. And then we waited for a certain time until we would harvest them and then look at the transcription factor binding sites. But how did we find out how much time did we have to wait for? So because the idea here was to see the differences between the different plant lines, because some of them are more resilient to drought and some of them are less resilient to drought. So what we were looking for was a point where some of them already were showing some symptoms of drought and were already having problems coping, but other plants were still looking really fine. So within this population, we tried to find this kind of sweet spot where there's a huge variation in the drought response. And we monitored that with different parameters, with some obvious parameters, which is something that maybe you're familiar with that, but maybe people have already seen that before. So when plants get drought stress, they roll their leaves. So maize plants roll in their leaves. You might see that in summer, sometimes especially at the edges of some fields. Then these plants, the leaves become really pointy and like rolled in. And this was one of the things we were looking out for. Then the maize plants also, they start to shut down photosynthesis, so they become less green at one point. But we also used some specific measurements. So we were measuring the water content within a specific part of the plant to really have some hard measurements where we could really have numbers and could put numbers on that. So this plant had this and this amount of this percentage of water still retained within the plant while the other plant had so much. And then we compared that to the plants which had been watered all the time.
Okay. And I think probably, I'm assuming an additional difficulty will be like the phenology of the different strains. Because like you say, we wait until plants have 4 leaves and then we stop watering them. But if one population reaches that stage faster than another with the same amount of water, then how do you fix that? Do you then say, okay, now we stop watering all of them, or you try to wait for the other strain, or how did you do that?
We actually waited. So we looked at each of the different lines that we had. In this case, actually, it's a bit more complicated. They were hybrids. They were crossed with a tester line. They were all crossed with one very drought-resistant line, so that we can compare them very well. But still, they had some differences in their development. And so for each of the lines, we went to the greenhouse every day, counting how many leaves were there, at which developmental stage they are, and then only when the line reached this point, we would put this one to drought and then harvest it the exact time later. So like after 3, 4, or 5, I mean, in one of the experiments, it was 86 hours, and in some it was 110 hours. It depends on the population somehow. So yeah, we would then have several different days to harvest them, which is nice because harvesting 30 of them would have been like really hard because it's not one plant that you're harvesting. We always have like a lot of different pots with a lot of different plants. So actually it was quite good to have that stagger.
Yeah, yeah, I can imagine it makes it more feasible in practice.
Yes.
Okay, so now we have defined drought. How do we proceed?
So what we did is we had the drought stress plants, and that at this moment when we decided, okay, at this point we want to really look at what's happening molecularly, we harvest the plants. So in that case, what we did is we harvested one specific leaf, so always the 7th leaf of the plant, and then in these leaves we analyzed the transcription factor binding, and then now we could compare, like first of all, between the lines. So are there differences which we can find, which are when you always see like, okay, there always when this base is mutated, then we see more binding or we see less binding. And then we compare that between drought and well-watered and saw like, okay, but this is a peak where we see differences between alleles, like these different bases, but we also see differences between drought and well-watered. So there is more binding, but also is there still differential binding, or is this difference becoming larger between well-watered and drought?
Yeah, so you compare between the different lines, but also between the control that is well-watered and the treatment that is being exposed to drought. And if you find these regions, then what do you do with them?
Yeah, so we found quite a lot of regions actually. When just looking at well-watered, we found like 150,000 regions, which
that's a lot,
which could be potentially interesting. But then what was very interesting is that when we're looking at the drought, we found like more than 45,000 additional regions, which we didn't find in the well-watered. So these were, of course, especially interesting. And then we used a lot of additional data to narrow down which ones are the most interesting ones. So we used, for example, the data of how strongly the genes are actually switched on, because you can measure that as well for the whole genome, so for all the different genes. So we checked if there were differences between like well-watered and drought, if the genes were more switched on the well-watered or more switched on the drought, and if that was correlated also with the presence of changes in the DNA or not. So we used that as one of the ways to narrow down what we saw, to find more specific candidate regions. And then we also used a lot of published data. So there are a lot of data sets out there where people already looked at which regions of the genome are somehow associated with drought. So without looking at transcription factors. So the disadvantage of this method, which is called a genome-wide association studies, is that usually you get like relatively broad regions. So normally it's not really known which exact part of the DNA within those regions is responsible for the changes that have been seen. But we have these regions of interest. So now we overlap them with our very high resolution data to see like which of our like 150,000, 120,000 under drought regions are actually located within these sites. And with that, we could really narrow them down. And then we are like in the hundreds or in the tens for some of these experiments with drought. And then that becomes a more handleable number to go further into detail.
Okay. So people have identified the broader regions for drought and you overlay those with your findings. Now you know which of your findings are interesting for drought. And now that you know that, what do you do that? Like, how do you proceed from that?
Yes, so that's where actually now the Booster Consortium comes back in. So especially our collaborators in Belgium, because what they can do is they use this, they can use the CRISPR-Cas9 technology, so these gene scissors, which I think you've made a podcast about that as well.
True.
So, and they can use this mechanism to cut out these regions in some of the maize lines, and then we will see if that really improves or actually makes the drought resistant worse in some of the lines that we're looking at. And now what the nice thing is we can then also combine some of these cut out pieces from different genes and see if they add up and make like a bigger difference when we would combine them.
Yeah. So the practical use would then be like now you will see with CRISPR which regions are actually affecting drought resistance, and then you will give farmers those lines that are more resistant, or what is the goal?
Well, that depends a bit on also on regulation within the EU, because you know that there's a big debate going on if CRISPR plants can be actually used within the EU. But luckily, we're also working together with breeding companies. And what they can do is they have a lot of populations already where they have crossed a lot of different plants with each other. And of course, they could also screen all of those for if they are more drought resistant. But if they have an idea of what they're looking for, they could actually screen those plants. Oh, do they have these regulatory elements which are actually good for drought stress? And then they could select only those plants and then look at them in drought conditions. And that's what people call like so-called smart breeding, which would then speed up the process of breeding. It's still, in a way, conventional breeding. So there is no transgenes involved and no gene scissors or something like that. It's just crossing plants with each other, but we would very much speed up this process to kind of get these beneficial alleles on the market.
Yeah, so the major difference in time is because of your research, they know which plants to use for their breeding.
Yes, and they look at which regions to use within their plants, which are maybe a genetic mosaic of some parents, and so they know at which plants to select from their populations.
Okay, so now I follow what you're doing, mostly for maize, but how does then Teff come back into the picture? So it's like you said, it's very drought resistant. So you want to transfer some of those genes to maize, for example, also with CRISPR, or how do you do that?
Yeah, so that in this case would then be this is more like an exploratory part, because that would then generate a transgenic plant, because we would put something from Teff into maize, unless we would learn something from Teff, which would be already almost there in maize. So we would just have to change a few bases. There's also a strategy which is called base editing, which is also not a transgene per se, but it's just like a gene editing technology. So that is something we have to decide once we have the results from the Teff, to see if there is like a lot of things to change to introduce these things into maize, or if it would be just like very few little changes, which could maybe done this way. But this is more like the exploratory part of that project. But also for the Teff itself, I mean, we can still improve. We think that there's still room for improvement for Teff. And the institute in Ethiopia is also a breeding institute. So they would be very, very interested in knowing which regions are really good to select for breeding. And they have had, so when I talked to them in Switzerland, they have had enormous improvements already with their breeding program, so to improve the yield on Teff. And yeah, they're super excited about having these sites and then using them for their breeding program as well. And we have also like another very curious plant that in the consortium, which we're not looking at the population, we're just looking at a few plants, but we want to understand how drought resistance works in this plant. And that is a plant from South Africa. It's called Lindensis. So it's like a resurrection plant. So it's one of these plants where maybe some people have heard of. They can look like really dead. So they're dried out for months and months. And then just when it starts to rain or when you water them, they kind of come back to life. And so we also want to look at transcription factor binding sites in those plants and see what's going on a molecular level with these plants. And if we can learn something from them, even for Teff or for maize, maybe.
Those plants are really insane. Like, I don't know the species you are talking about, but I had a friend who did research on resurrection ferns. They really look dead, and then you give them some water and they're just back. It's fine. It's all okay.
This is more like a grass, but yeah, exactly the same. So they look dead and then they come back.
Yeah. Well, it actually ties back a bit to your fun fact from the start that plants can live forever.
Yeah, that's true.
If they have the right equipment.
Yeah.
Now, from this project, you already have some findings that you're able to share with the listeners?
Yeah, so one interesting finding was that, coming back to this idea that you always have trade-offs with drought-resistant genes and so on, we found a lot of our interesting binding sites, which also correlate with these genomic sites, which have been found in other studies, in a gene which has already been studied in the model plant Arabidopsis. And in this model plant, this gene is called tiny. And what people found is, like, if you switch on this gene in a very strong manner, then plants are very drought resistant. But this gene is also called tiny for a reason, because these plants are really, really small. So if you overexpress or if you make more of this gene, then yes, they're super drought resistant, but really, really small. So, and there is a homolog, so a kind of gene in maize, which is very similar from its sequence to tiny. And what we saw there is that, yes, first of all, it was found as a region which could be interesting for drought resistance with the genetic variation, but also that there is a region which was found to be involved in leaf length and in growth of the plant. So it seems like this gene is doing very much the same as in Arabidopsis. So it's regulating growth, but also drought stress. And within this gene, we found a lot of these interesting candidate binding sites with our method. And what we also found is that within our population, we really have some of these cases where this gene is like still switched off under well watered, but then under drought conditions, it gets very strongly upregulated. So we have some alleles where it really looks like we can find exactly this type of regulatory elements what we're looking for, where we're like, they're not on under well-watered conditions, so the plant is not making a lot of additional protein, it's not wasting its resources, but then when it's needed under drought stress, this can be switched on. And this is one of the genes where we're now looking at with our collaborators which they're studying in more detail. But also we've looked at isolated maize sets, and we've simulated some drought stress where we just put a small region with these kind of binding sites in front of something which is called a reporter gene, so which can tell you if this small piece is actually switching on a gene or not. And we simulated drought stress by giving those plant cells the hormone, which is responsible for signaling drought stress within plants. And we could really see that when we have this base sequence, which is also in nature, is like correlated with really strong switching on of the gene, that also happens in this kind of simulated environment when we only have a smaller part of the DNA and not the whole genome. So this is one of the kind of, really promising candidates. We have a few more, and we're now working on figuring out more how well they can actually be used to improve drought stress.
Yeah, well, that already sounds very promising. So if I understand correctly, the gene that is regulating drought stress also makes the plants smaller. But I think One of the challenges is probably that you don't want the plant to be small also when it's well watered. So you want it can reduce growth when it's drought, but not when it's well watered. Is that the main challenge for your research, or are there other challenges as well?
This is one of the challenges. I mean, that's what we want to solve in the end. Among others, I mean, there could be also, we're also looking at some genes which, for example, are switched off under drought stress. So they might just be genes which, if you switch them off, they would be beneficial. So it works also the other way around, and we're also looking at that. But another challenge that we're facing is that it's not always clear with these regulatory elements, and we might find some very interesting ones, but we don't always know to which gene they belong, because as we saw in the beginning, they can be like really, really far away from genes. And it's not always evident with which gene they are working with or which gene they are regulating, if they are regulating a gene, because a gene can also have a lot of different regulatory elements. So even if you're comparing two plants, so one could have like a negative element and a positive element, and the other one could have them changed, flipped, then you see the exact same amount of protein being made, but the elements have been changed completely. So this is a huge challenge that we're facing, and because of that, we are trying to look into long-range interactions, so into looking like into these kind of loops of the DNA I was talking about in the beginning, so where we can see that maybe some parts of the DNA can bend around and can come into close proximity to a gene. Although if you look at the linear DNA, it's very far away. They are very far away from each other. So this is one of the challenges that we want to tackle with our new projects. Go on with that.
Okay. And if everything goes well with this project, when will the results, or actually the practical results, like the seeds, the plants, be available for farmers?
Yeah, so in the booster project, I think we estimated that it might be like 5 to 8 years after the end of the project. So that's in like in 2 years. So it would be like in between 7 and 10 years when that would be like going to the market, which is already quite fast, I think, in this market, although it sounds quite a long time.
Yeah, but I think maybe that's a good point to make, that it can take a long time before all these things progress and also get approved to get to the market and stuff like that. So that's, I think, a normal and realistic timeline. So you actually touched upon it already a bit, but are there like other steps you want to take in the future with your research?
So at the moment, what I want to do is that we want to transfer the methodology to other plants. So I mean, now we've been looking at maize and some relatives of it, but there are a lot of different crops out there. And as I said, we are more looking into tropical crops now, because they have a high chance to be already quite drought resistant. So they are very interesting for like having the potential to find these kind of natural variants which are already drought resistant. And with that, I'm, for example, looking into pearl millet, which is also a plant which is very interesting for Africa. It's cultivated a lot over there. But these plants can already also become very interesting for Europe, because with the changing climate, they might also be like quite beneficial here. So that's one of the parts, like transferring the method to other plants. Then, yes, as I said, the next step would be like looking at these long range interactions. And then another step would be to look more into the data side and kind of get AI researchers involved. And at one point, the dream, of course, of everybody in the field is that at one point, we don't have to do the experiments anymore. We just have to look at the DNA sequence and we can make predictions. I mean, that's, of course, very far in the future, but that's something what we're working towards with some collaborators and getting and going into this direction.
Yeah, that would mean that we completely understand what every base pair, more or less, of the DNA is doing. Okay. Are we far away from that, you think? Like, how far away are we?
Yeah, so it's very hard to say, but I think we're not so far away for certain characteristics. So for like example, for a transcription factor where we know which DNA sequence it likes, And we've already seen that we can train models to then predict where these transcription factors are binding. But okay, that's like predicting the molecular events. That's something which is, I think, relatively feasible in the near future. But then I don't know how close we are into predicting phenotypes. So we're working on that, and maybe it will work like for part of the phenotypes and not for everything. But what you said, just like understanding what every single base pair does, I think we still have a lot of work to do.
Well, that's good. Otherwise, we would be out of a job, I think.
Yes.
Okay. Now, before we close, do you have a take-home message for our listeners?
Well, other than that, the plants are really cool and we can really learn a lot from them. I think we can learn a little bit from this idea that people thought that the non-coding genome was junk DNA. So it really tells us that we shouldn't dismiss something that we don't understand as junk. We should just keep our patience, look into it, and then find out what it's doing. And just stay curious and find out what these things are for and not dismiss them so easily.
Perfect. I think that's a good idea for science, but for everything in general. Don't think things are junk. They might be interesting.
Exactly.
This was Apple Finch Pudding. I want to thank Julia Engelhorn for the information. Let's meet again for the next episode of Apple Finch Pudding.